So in this lecture, we are going to start talking about enzymes, and we're also going to discuss the coenzymes and their function in the reaction. So basically, biochemistry begins with enzymes, and enzymes, of course, are going to be proteins. So they're going to be encoded in our DNA. Now, an exception of this rule is that we now understand that there are ribozymes, which are made up of ribonucleic acids that can act as enzymes. And they are, in essence, not proteins because they are RNA catalysts. And we'll talk about a few of those in coming lectures. Also, enzymes, of course, are going to be significant for life. Life depends on enzymes' activity. And here is just a picture of a hypothetical reaction. And after 1 year in the absence of an enzyme, this little dot or this little area brown here is all the reaction that took place within this hypothetical situation. In the presence of an enzyme, after 1 second, now we have a lot of reactions occurring. So this is what enzymes do. Enzymes are going to speed things up. Now here are some just basic terms that go hand in hand with enzymes. So we have a prosthetic group, and a prosthetic group is a coenzyme or a metal ion. And these are going to be tightly bound to the enzyme. Okay, now a holoenzyme is going to be the active enzyme. And so the holoenzyme is going to be the enzyme with its bound coenzyme. And then an apoenzyme is an inactive enzyme. And this is going to contain the protein part of the holoenzyme, in essence, the enzyme. Okay, so some metalloenzymes are going to be enzymes that are going to bind to and retain metal atoms with high affinity. Okay, so now we have a list here of inorganic ions, which are referred to as cofactors. So just note that a cofactor is any non-protein substance required for an enzyme to be active. So in essence, our coenzymes, cofactors, are not encoded in our DNA. And so these can be organic or inorganic. And so here's a list of inorganic ions that interact with our enzymes. And so listed here is going to be our ions. And then, for example, iron is going to be important for cytochrome oxidase, for catalase and peroxidases. Our potassium is going to be important for pyruvate kinase. You have your magnesium, you have nickel for urease, zinc for carbonic anhydrase, alcohol dehydrogenase is going to use zinc, etc. So all of these inorganic ions are going to be part of the enzyme, and more specifically in the enzyme active site. Now, enzymes are part of cofactors, but they are going to be organic cofactors. And in this case, we have our coenzymes, and most of them are going to come from our vitamins. So we have biocytin, which comes from biotin. We have, of course, coenzyme A, which comes from pantothenic acid. We have our flavin adenine dinucleotide, or FAD, that comes from our vitamin B2 or riboflavin, etc. So all of these coenzymes you're going to see pop up over and over again with different enzymes and chemical reactions. So just very quickly, very briefly, when you name enzymes, you add an ACE to the name of the substrate. And typically, an enzyme was going to be named for their function before the specific chemical reactions were known. And then they were named for the source of the reaction. And some enzymes ended up having multiple names. And so they were identified by different people, given multiple names. And in the literature, that got very confusing. So now there's currently an international agreement for naming enzymes. And we've already talked about this international agreement. So they're given a number and then a classification associated with that number. So if it's an oxoreductase, it's given a classification of a number 1. And these, of course, are going to be important on adding or removing hydrogen atoms. 2 is our transferases. They're going to transfer functional groups. We have hydrolases that are going to add water across a bond. We have lyases that can add water, ammonia, carbon dioxide across double bonds. They can also go in the reverse direction. Isomerases are going to change the look, so going from D to L, moving around different elements within a molecule. And then ligases are going to join. You're going to join 2 different groups. Now, coenzymes are important because they help make things happen, and in fact, many enzymes could not function without their coenzymes, which is why vitamins are very important in our diet. So coenzymes are going to be necessary, and usually they're going to be used by 1 or more different enzymes. Now also they're going to be present in a small amount, and this is in comparison to the actual substrate. And they also get reused. So they're going to be regenerated and reused over and over again. Now enzymes are going to exist free in a solution, most of them. And then they're going to have some that are going to be bound, like those prosthetic groups we just talked about. And then they're going to act as transporters of a chemical group from one reactant to another. And so they're basically going to carry groups, and they're going to donate the groups. And so you'll see that our coenzymes are going to bind not only to the enzyme, but they're also going to bind at a different site to the actual substrates. So in general, there are 2 main categories of coenzymes. They're called activation transfer coenzymes or oxidation reduction coenzymes. Now we'll talk first about our activation transfer coenzymes because they are going to have 3 main features. They all have a specific group that's involved in binding to the enzyme. And then they're going to have a separate and different functional group that participates in a catalyst by forming a covalent bond with the substrate. So you can think of it as your body, and you have 2 arms. One arm is going to grab, or one hand of your arm is going to grab the enzyme, and the other hand on your arm is going to grab the substrate. And then your arms are flexible, so they're going to bring those 2 together to cause some sort of reaction. In addition, they all are going to have dependence on the enzyme for specificity of the substrate. So they can bind to the substrate, but in order for the chemical reaction to go forward, that substrate has to be able to fit and bind into the active site of the actual enzyme. The enzyme also is going to provide catalytic power, of course, so the coenzymes bring things together. So the speed at which a reaction goes forward is not only the presence of the enzyme, but presence of the coenzyme being able to bring the two entities, enzyme substrate, together. So one example of this is thiamine pyrophosphate, or TPP. And TPP is going to form a covalent bond to part of the substrate, and then it's going to also interact with the enzyme. So the other part of the substrate will bind to the enzyme. So in TPP, you have an amino pyrimidine ring. And that's shown here. You also have a thiazolum ring with that thiol group. Okay, and then you have two R groups. And so each of these are, those amino ring is one R group, and then you have a second R group. Now, thiamine triphosphate is going to interact with the enzyme. And the part that's going to interact with the enzyme is going to be the hydroxyl alkyl groups. And so your enzyme is going to come in and grab the hydrogen that is next to that thiol group. And so that hydrogen is now going to be able to dissociate charges, for example. And so your TPP, shown here, is going to be involved in the decarboxylation of pyruvate, and it's part of the pyruvate dehydrogenase complex. So here we have pyruvate. Pyruvate is a 3-carbon compound, comes from the glycolysis. Use a highlighter instead. So here's our 3-carbon compound. And this 3-carbon compound is going to be converted to a 2-carbon compound. Eventually, not in this first step, but through the pyruvate dehydrogenase complex. But when interacting with thiamine pyrophosphate is going to be able to have a strong nucleophile because of that hydrogen that was removed. And so here's our strong nucleophile again next to that thiol group. And the strong nucleophile is going to be able to bind to the carbon with a partial positive, and that's going to displace carbon dioxide. So you're basically removing carbon dioxide. You're going from a 3-carbon to a 2-carbon, and you're releasing that 3rd carbon as carbon dioxide. And so your thiamine pyrophosphate is going to help to stabilize that removal of carbon dioxide to give you that 2-carbon result. And then you have resonance that goes between 2 different species between the 2 different TPPs. Now we have our R groups, of course. I should do one in a different color. And so we have the R groups are still part of this TPP. So once TPP releases the 2 carbons, which it eventually will do in pyruvate dehydrogenase complex, it will regenerate TPP so it can work on another molecule of pyruvate. Now another example of an activation transfer coenzyme is coenzyme A. Now coenzyme A is a very important 2-carbon build for adding 2 carbons. So it's important for 2-carbon building block. So it's going to form thioesters with acyl groups, and here's our acyl group shown here. And you're going to have an acetyl group form, or a succinyl group, or fatty acids also will be involved with coenzyme A. Now, the acyl groups are usually going to be derived from carboxylic acid. So that's going to derive your acyl groups. Acetyl-CoA's main function is to deliver the acetyl group to the TCA cycle. And this, of course, will make energy. Now, if you look at what's going on here, let me raise this a little bit. You have this region which is going to bind to the enzyme. And then you have this thiol group again, which is going to bind to the acyl group. Within this molecule, you also have an amide link, which is here. And then so this thiol group, make it a little bit bigger because it includes the amide group, is basically from cysteine. So cysteine's going to donate that sulfur to attach to the acyl group. And then so your enzyme, or your vitamin, your coenzyme is made from pantothenic acid, which is vitamin B5. And this together is going to facilitate the transfers of acyl groups to a substrate. And so your enzyme is going to bind at that phosphate, and your coenzyme is going to help to bring your enzyme and that acyl group together. Another activation transfer is biotin. Now, biotin is going to be very different. Biotin has no phosphate group. Now, it does not mean that ATP or one of its derivatives is not part of this reaction, but it's the coenzyme itself does not have bound phosphates. Now, the other thing is your biotin always has to be bound to a lysine on the enzyme. And then biotin is going to be able to pick up a carbon, and it's going to be in the form usually of bicarbonate. And it's going to pick up a carbon, and it's going to transfer the carbon. And it's going to be a carboxylation reaction. So one thing to think about, so here's a note, is that we have our dehydrogenase, which can take away a carbon. And we have our carboxylase. And this can add a carbon. And both are going to be very important, and both, if they occur, are not reversible. So if you take away a carbon, it's not the simple reversion of that chemical reaction to add it back. And the same is true for a carboxylase. If you add a carbon, you can't just go in the reverse. You need another enzyme in order to do that. Okay, and then we have pyridoxal phosphate. And again, this is another one that does have a phosphate attached to it. In this case, your aldehyde, shown here, is the functional group. This is going to form a covalent bond with the amino group of amino acids. And so this is referred to as a Schiff base. what is the result is that you get a charged pyridine ring. And this pyridine ring will pull in electrons. And so this equals an electrophilic catalyst. Okay, and so basically your Schiff base, and this is vitamin B6, you have your amino acid. Let me do this in a different color. You have your amino acid that's going to come in and it's going to react with the aldehyde. And then this is going to, so this is step 1. Step 2 is that you're going to get that ring formed. And then on that carbon up here where you had your aldehyde, now you're going to get your amino acid bound. So here's your amino acid. And then of course you still have your phosphate. And then here you have a few double bonds going on, and you have your nitrogen. And so you can get a shift of charges through your nitrogens. And this will be a Schiff base. It's an intermediate towards finishing the chemical reaction. But this is where a Schiff base comes from. Right now, so the next type of coenzyme is an oxidative reduction coenzyme. And this is going to be involved in oxoreductases. And so we have NAD and FAD that are referred to as coenzymes. Even though they typically don't regenerate themselves directly after a chemical reaction, they are taking electrons, but eventually they do get converted back after the electrons are donated. So they can transfer electrons. Are going to be important in making ATP. We also have the important part of these guys is they have no covalent interaction, no covalent bonds with the coenzyme and substrate. Metals can also be involved in these oxidation reduction reactions, and they transfer single electrons to oxygen. And so it's very important to control metals because you don't want to generate reactive oxygen species. You're going to have vitamins E and C, which are going to function as coenzymes similar to activation transfers, but without chemical bonds. And then vitamin E itself is going to be an antioxidant that protects from free radicals, so protects from those potential reactive oxygen species. Now, an example of this is NAD. NAD is a vitamin niacin with ATP. An example of a enzyme that's going to use this is going to be lactate dehydrogenase. And this is going to transfer electrons from lactate to NAD. So you have lactate here, and it's going to donate this hydrogen, which is essentially an electron, and is going to donate it to NAD. And then your functional group in your nicotinamide ring is the carbon. So your functional group is going to be this carbon here. And then your hydrogen from the substrate is going to dissociate, and a keto is going to form, a ketone. And then NADH is going to dissociate. Now the other thing to note is ADP portion of NAD is going to bind to the actual enzyme. Okay, and then this, of course, lactate is the substrate. And then NAD dissociates, and then NAD can donate its electron, or it releases the electron and forms your pyruvate. Now, some of the similarities between both of these are that each are going to have a group that accepts and donates electrons. And then there's going to be a different part of the coenzyme that binds to the enzyme and a different that binds to the substrate. And they are not good catalysts without participation from the amino acid side chains of the enzyme. Now, differences are that the activation transfer form covalent bonds. And this is with the substrate. And your oxidation reductions do not. Now let's just talk very briefly about metal ions. So metal ions are going to have a plus charge. And so they're going to be electrophiles. In that they attract electrons. Now they're going to assist with binding a substrate, or they can stabilize anions. And so they can accept an electron, or they can donate an electron in an oxidation reduction reaction. And some can bind to and participate in binding of substrates or coenzymes to the enzymes. Should be bind. An example is your magnesium is going to bind negative phosphate groups. Of that TPP that we just talked about, and also to anionic or basic amino acids in the enzyme. So magnesium will bind both to the coenzyme, which is TPP, and to amino acids in the actual enzyme. Another thing to note is ATP molecules always are associated with magnesium. Even though it's not written down that it is associated with magnesium, it is. Now, in this site over here, we have our alcohol dehydrogenase enzyme. And in our alcohol dehydrogenase enzyme, we have a few things going on. So the first thing is we have serine. So in step 1, we have a serine, and serine pulls a proton away from ethanol. Okay, and then in our second step, we have a charge on the oxygen, and the charge on the oxygen is stabilized by zinc. And then this is going to allow for number 3, the transfer of our hydrogen to our NAD. Our electrons. And then NADH will dissociate, and then you will be breaking down ethanol or metabolizing ethanol. Now you can also see in here that we do have a histine, and our histine residues are usually involved in deprotonation reactions, and they're also involved in helping to stabilize the electron movement within the active site of this enzyme. Now, cofactors can also be non-catalytic, and they can bind to enzyme tertiary structures, or they can serve as substrates. They can be cleaved during the actual reaction as well. And then finally, let's just talk briefly about heavy metals. So heavy metals are associated with toxicity. And so we can have mercury toxicity, we can have lead toxicity, we can have aluminum, or iron are very common. And so all of these are going to bind to the functional group of an enzyme. And so these are going to be very nonspecific. And so, for example, your mercury poisoning will bind to sulfur hydro groups. And so they bind many enzymes. They're going to disrupt many interactions. So our mercury binds to sulfur hydro groups. Your lead is going to replace normal metals in enzymes, and this is important in developmental and neural toxicity. Some of this is thought to be because lead can replace calcium in the CNS, and it interacts with calcium calmodulin pathway as well as protein kinase C pathway. So in essence, though, it's hard to tell exactly what is causing the disease because heavy metals can have an impact on many different chemical reactions in the body.